complex organ existed which could not possibly have been formed by numerous successive slight modifications, my theory would absolutely break down, adding, but I can find out no such case. In this, Darwin was emphasizing that his mechanism of random mutation natural selection had to improve life gradually in tiny steps over long periods of time, because he knew that if life improved in big leaps suddenly, it would look very suspicious, as if non-random processes were involved with that. Well, one problem, Darwin's theory has been around for a while, and nobody's really answered his criticism. And one problem is that most people actually think, like Darwin did, of evolution in terms of organisms. Okay, fish with legs. This guy is being harassed by his wife. Don't walk away when I'm talking to you. And here's a dinosaur sporting feathers that's been in the news. That's what people tend to think about. But it turns out that modern science, its biggest claim to fame, I think, is the discovery that the basis of life is actually molecules and information and molecular machinery. The cell, the operation of the cell, was unknown to Darwin. Scientists of his day thought that the cell was filled with jelly, protoplasm. They thought it wasn't a big deal at all, just a simple little jelly. But now we know that, in fact, it's filled with DNA and molecular machines that we'll see, and that the sequence of the DNA carries real information, just like the sequence of letters in a book carries information. And so that's made that's really elucidated life and made it easier to evaluate Darwin's theory. So let's do that. I did that in my first book. Darwin said that if something couldn't be produced by numerous successive slight modifications, it would be a big problem for his theory. Well, what sort of a system, what sort of an organ could not be produced, or sure looks like it couldn't be produced, gradually? And the kind of system that challenges Darwin's mechanism on that point is one which I call irreducibly complex. And it's a fancy phrase, but it stands for something simple. It just means you've got an organ or a machine or a system that has a number of different components, and they interact with each other to produce some effect that the individual components cannot achieve. And if you take away one of the components, the system doesn't work anymore. So it's irreducible because you can't take anything away, and it's complex because it's got a number of components. And a good example, I think, of such a system from our everyday run-of-the-mill world is this, just a little mousetrap, a mechanical mousetrap. It's got a number of parts. It's got this wooden platform to which everything else is attached. It's got this tightly wound spring with an extended end that pushes against the platform on this side, and in this is pressing against another metal piece called the hammer, which is the piece that flips over and squashes the mouse when it trips the trap. And it's got a number of other components as well. And these all have to be precisely matched and all together. If you are missing the spring, or if you're missing the hammer or the platform, it's not like the trap catches mice half as well as it used to, or a quarter as well as it used to. It's broken. It doesn't work. It's surprisingly difficult to think of a route by numerous successive slight modifications to some primitive trap that would yield a trap such as that pictured here. And in the 20 plus years since the book came out, there's been a veritable internet cottage industry in trying to construct such things. And in my utterly unbiased opinion, they all fail completely. We could talk specifics if somebody wants to. Well, okay, so there are such things, and if a mousetrap is irreducibly complex, and it's a pretty simple system, then anything much more complex than that is going to be it too, or very likely to be. Well, are there any such systems in biology? And it turns out that yes, they're all over the place at the molecular level. One big discovery of biology in the past 50 years is that the cell is run by machines, literally machines made of molecules. Has anybody here ever watched a TV show called Star Trek? Okay, yeah, a couple people have. Well, in one of the later versions, there's these creatures called the Borg, which are half humanoid, half machine, and they're run by nanotechnology. Well, in a very real sense, you are the Borg too. You're run by nanotechnology. You're run by machines in your cell. And here's a good, I think, visual example of one molecular machine. It's from a bacterium. It's called the Bacterial Flagellum. And the flagellum is literally an outboard motor that cells use to swim. Just like a real outboard motor, this part rotates in a circle, and this filament-like thing acts like the propeller. It pushes against the liquid that the bacterium is in, and therefore pushes the cell forward. It's attached to the drive shaft by a region called the hook region, which acts as a universal joint. The drive shaft is attached to the rotary motor. The motor has a stator that keeps it in place as it's rotating. There's bushing material to, There's bushing material to allow it to pass through the membrane. And it has dozens of parts, and if you take any of them away, it doesn't spin half as well as it used to, or quarter as well. It doesn't spin at all. And this is actually just a little cartoon version. It's like representing a computer by a little square box with a line connecting to another little square box. All of these components that we see as simple geometric shapes are themselves very, very complex. So most scientists will say, yeah, you're right, the molecular level of life is extraordinarily complex. And yes, there are things like this, and we have no explanation for how they could be produced. But I part company with them when they say things like, well, we'll wait around and see what turns up. I say that if you look at the flagellum, you can tell that this is a machine. It's not like a machine. It's not analogous to a machine. It's a real machine, and we know where machines come from. The flip side of, we know that machines require design. And it turns out that we recognize intelligent design when we see that parts, physical parts or other kinds of parts, have been arranged to match each other to produce some effect that otherwise couldn't be produced. Something like irreducible complexity. Well, let me give you a little illustration of that idea. It's shown on the next slide here. And this cartoon is a Far Side cartoon. I love Far Side. And you got a bunch of jungle explorers and one that has been strung up and skewered. And this fellow turns to this guy and says, "That's why I never walk in front." Now, everybody here, and earlier this morning, if you were there, you look at this and you immediately realize that this was designed. That's not an accident. You realize that this had to be put together by an intelligent agent somehow. As a matter of fact, the humor of the cartoon depends upon you recognizing the design. It wouldn't be real funny if he, just stepped on a snake or something. Okay, so I argue that not only that irreducible complexity is a double-edged sword. Not only is it a barrier to Darwinian processes, but it also is a very strong pointer to purposeful design, because irreducible complexity is defined as when parts are essentially matched to each other to produce a function, just like this. And that's what intelligent design is. Okay, so that's what I pretty much said in the lecture this morning. So let's continue from there. And about 11 years after the first book, I wrote a second book called The Edge of Evolution. And the gist of it is that, suppose you're like me and you think that some things in life require design. You can't get a flagellum unless you explicitly make it. But not everything in life requires design. There are genetic defects, and they're inherited, and, the shape of my nose, there's no reason to think that was explicitly designed. So if you think that stuff in life, some stuff requires design, but not everything requires design, where is it reasonable to draw a line between what you think did require design and what might have occurred by some random accidents and selection, a Darwinian mechanism? And I call that line the edge of evolution. And that's what the rest of the talk will be about. So let's ask that question. But first, let's start asking ourselves, what do we mean by Darwinian evolution? What is Darwinism? Well, it turns out that it's not a single theory. It has a number of parts. And if it has a number of parts, then some of them might be correct and some of them might not be correct. So, for example, one part of the theory is the idea of common descent. That is, that organisms living today have descended from organisms living in the misty past by birth and death. And if you think about it, common descent, well, that's interesting. It's an interesting statement about nature. But even if you think it's correct, it's pretty much trivial, because it doesn't say where the ancestors came from. It doesn't say how they turned into what we see in modern life. So it doesn't do anything except say that, well, what this organism has is similar to what that organism has, so maybe it descended from a common ancestor. Well, okay, that's interesting, but again, I think it's kind of trivial. Another portion of Darwin's theory is natural selection. And of course, that's survival of the fittest. The better adapted organisms will survive. And again, that's interesting in my view, but kind of trivial. I mean, who's going to argue that the more fit organisms won't do better on average? That if some organism is bigger and bigness helps in your environment, that it won't fare better than smaller organisms. That's pretty straightforward. A third idea in Darwin's theory is the idea of random variation, or these days, random mutation. And this, in my view, is the critical claim of Darwin's theory. With this claim is all 99% of the philosophical and even theological and other and scientific weight of Darwin's theory, that the mutations which feed natural selection, the changes which occur, are random. They just happen. They happen by physical processes that no one directs. So it is this branch, this idea that's packed into the multi-part theory of Darwinism that I'll be focusing on tonight. Well, okay, so how do we test that? Well, one way you can test it is to ask yourself, well, what do we see Darwinian processes doing? What's our best example of what a Darwinian process can do? And at the time, in the mid-2000s, the best example of Darwinian evolution was malaria. Now, malaria, you may remember, is a terrible disease. It infects 100 million people a year. It kills 1 million people a year. Even now, it's transmitted by mosquito, but it's not the mosquito itself that is the disease. The mosquito carries a single-celled parasite, the malarial parasite called Plasmodium falciparum. That's the worst of the breed. And what happens is that the single cell gets inside a person by being injected by the mosquito, and it goes to the liver, and it hangs out in the liver for a little while, and it replicates there. And after a week or so, about 20 or so cells come out and go into the bloodstream. And while in the bloodstream, a malarial parasite attaches itself specifically to a red blood cell. And it uses machinery to enter into the red blood cell. And it eats the hemoglobin found in the red blood cell. And it uses the energy it derives from that and the materials to make copies of itself. And it makes about 20 copies of itself in each cell, and then it bursts open the now trashed red blood cell, and each of the 20 hook onto another red blood cell. And they reproduce and burst open. And the next round, 400 come out, and then 8,000, and so on. It goes up exponentially until a person, half a person's blood is consumed, and that's what can kill a person. And a person sick with malaria can have up to 1 trillion cells, malarial cells, in his body, 1 trillion of them. And since about 100 million people get sick with malaria every year, that means there are 10 to the 20th malarial organisms each year on the earth. An astronomical number, huge number. Well, it's historically been a very serious disease, malaria has. But after World War II, a drug was invented called chloroquine, which killed malaria, the malarial parasite. A person could take a tablet or two of chloroquine, and magically, it seemed, they were cured after a day or so. It was great. And everybody in the scientific community was optimistic that we would eradicate malaria, just like, say, measles or smallpox was eradicated. But it was not to be. And here's a headline from the journal Science from about 10 or so years ago: a requiem for chloroquine. Turns out that the drug was effective for about a decade or so, but then it started to happen that physicians would notice some people would take the pill and they wouldn't get much better. And they'd have to do something else, and then they'd be cured. But then the next time, they'd get sick and they wouldn't be cured. And the malaria was developing resistance to the drug. And it did, and the resistant strains spread throughout the world, so that now chloroquine is pretty much useless for treating malaria. Well, it's interesting. It's very interesting, because if you look at other drugs used to treat malaria, you get much different results. There's a drug called atovaquone, which some people discovered in a lab. They had a little culture of malarial cells, and they put in this drug and killed them in a flash. So they say, hey, let's use this. So people would use it, and instead of a decade to develop resistance would occur in about every third patient who took it. So it didn't even require a week. So resistance to atovaquone arose in every third patient or so, which is, as I said, about 1 trillion cells, because malaria reproduces, you get about that many cells in a single person. Yet resistance to chloroquine arises in every billionth patient. It takes that many people before a resistant strain of chloroquine is found. That's about 1 in 10 to the 20th cells or so. So people noticed this, but nobody knew exactly how. Nobody knew what was going on. And in my book, The Edge of Evolution, I propose that, a straightforward explanation for why chloroquine was resistant why malaria couldn't develop resistance to chloroquine while it could to atovaquone and other drugs, is the same thing that makes it hard to win the Powerball lottery. Okay? If you had to match a number from 1 to 100, say 22, what are your odds of getting 22? Well, it's 1 in 100, okay, because that's how many other numbers there are. Well, how about matching two numbers, 1 to 100, say 22 and 51? Are the odds against it 1 in 101? Or are they 1 in 200? No, they're multiplied. It's 1 in 100 times 1 in 100, or 1 in 10,000. And the more and more you have to match, the more and more it goes up exponentially. You have to multiply the odds against each other. And I said, if the odds of getting a mutation in malaria are roughly 1 in 10 to the 10th, which sort of kind of matches the 1 in 10 to the 12th that takes to get a mutation resistant to atovaquone, then suppose you need 2 mutations before malaria acquires resistance to chloroquine. And I thought it was pretty straightforward, and I went on to develop an argument that I'll tell you about because of it. But Darwinian biologists laughed and slapped their knee and said, "Oh, that's ridiculous, that's silly, after all this, we know this guy's an intelligent design proponent, we know, such a thing would not happen." But it turns out that it does, and later work identified 2 mutations that were required to occur in a particular protein that resides in malaria. And when you change these amino acids, it turns out that it lets chloroquine leak out of the place where it usually does its damage in the cell. So the point is that if you need 2 mutations, it's a quantum leap from just needing 1 mutation. What would happen if it took 3 mutations to confer chloroquine resistance, on malaria? If that were the case, it would take 1 billion years or so for the parasite to get resistance. And that would be terrific. So the argument I make is this. Malaria had 10 to the 20th chances to come up with a resistance mechanism to chloroquine. That's how many cells were born and died. 10 to the 20th. That's, like I say, it's an astronomical number. It's more stars than there are in our galaxy. And yet, what did it do? It changed a couple of crummy amino acid residues in a pre-existing protein, a couple of what are called point mutations. So this, did it make a new, say, flagellum? Did it make a new molecular machine to efficiently get rid of it? No, it just tweaked what it had around the edges. And I said that this is not an argument that Darwinism cannot make complex functional systems. It is an observation that it does not. Because we weren't controlling malaria in the field, it could have done whatever it wanted to. If there were something else, an accessible pathway whereby it could become resistant to malaria, doing something else, modifying something else, coming up with some completely different machine, who's going to stop it? But in 10 to the 20th chances, the best it could come up with was, like I said, a couple crummy point mutations. So in fact, there is a good reason to think that once you need more than one change, if you need more than one change, a couple coordinated changes that work with each other, then you start to go well beyond Darwinian evolution. Malaria could overcome that challenge because it has huge population numbers. It has, essentially, it can play the Powerball lottery 10 to the 20th times. With larger animals like, say, mammals or so on, they have much, much lower population numbers, and you would never expect something like that to happen in a very, very, very long time. And let me just emphasize that point, that not only didn't Darwinian processes achieve anything much, but no unintelligent process helped much with malarial chloroquine resistance, including Darwinism, self-organization, self-engineering, symbiosis, which are all other things that biologists have talked about, nor any as yet undiscovered process. Because, like I said, who could stop malaria from doing what it could in the field? If there were something that was more probable than those two mutations, it was free and it would have come up with it. The odds say that you must come up with it. It's like saying, playing a Powerball lottery and you never come up with you never match one digit or something like that. If you play a very large number of times, you would expect at least to match one digit. So you can tell what is excluded by the number of chances that the organism has been given to try to overcome it. Now, I tried to take this argument and kind of run with it. I quoted some guys named Jerry Coyne and Alan Orr, and they wrote a book called Speciation a while back. They said that the goal of theory is to determine not whether a phenomenon is theoretically possible, but whether it is biologically reasonable. That is, whether it occurs with significant frequency under conditions that are likely to occur in nature. So if you talk to Darwinian biologists and point out problems, they'll say, I can think of something, and I can think, theoretically, this could go over here, and that could go down there, and why? What's the problem? You end up with a molecular machine. But the results, I say, with malaria show that's not biologically reasonable. If you need more than just a couple amino acid changes, you're in trouble. And I continued saying that, well, heck, a lot of things in biology would be expected to require more than one mutation to form. For example, you know that proteins are the basic components of molecular machine, and they stick together through protein-protein binding, where you get positive charges next to negative charges, and hydrophobic residues next to other hydrophobic residues, and have complementary surfaces that can kind of stick to each other like two magnets. This is an illustration of such a thing where this bigger shell is supposed to be a second protein. This is a smaller protein, and not only is it geometrically complementary to the first one, it's also chemically complementary in these groups, which I won't go into. But typically, you would need, a handful of mutations to make a reasonable binding site. But if you need a handful of mutations, the results from malaria say that would be virtually impossible for any reasonably sized population. If malaria with 10 to the 20th cells can only manage 2, what if you needed 3 or 4 to get such a site? Well, if protein-protein binding is beyond the edge of evolution, which I argued for, actually I argued that 2 protein-protein sites would be well beyond the edge of evolution, then we're in big trouble, because almost all of the machinery of the cell is composed of many protein-protein interactions, at least 6 or 7 different kinds of proteins that are arranged so as to give structures that can do very specific functions. This thing here is called a cilium. It's a very complex molecular machine, and each one of these colored dots that you see are copies of different proteins. The blue ones are a particular kind of protein, the light blue ones are a different kind, the white ones, the yellow ones, the green ones, and so on. Again, this is just a cartoon, but things like this would be well beyond the edge of evolution. So, some people say, well, suppose you already had a protein, how difficult could it be to develop a protein-protein binding site? Well, think of it in terms of a jigsaw puzzle. If you already had a protein, then it would not represented by this rectangular shape, and it would somehow be beneficial or selective to interact with another shape, another couple shapes representing proteins. You would first have to modify their shapes so that they were complementary to each other, so that they could fit together into a molecular machine. And with the results we already have, that's extremely unlikely. I went on then to say, well, so what's the edge of evolution? And again, most people aren't interested in cilia and flagella. They care about bunny rabbits and elephants and organisms you can see. Well, I argued that, in fact, if you look at nature, then design or very fine tuning can be seen from the very foundation of nature, that is to say, the laws of nature and physical constants and so, through things like the origin of life, down into the cells and genetic codes to molecular machines, and then I started to talk about biological categories, biological kingdoms, phyla, cell types, classes, and I said we have information down to classes that they are very likely to require design. And I argued that because classes are things like fish versus birds versus mammals. The thing is that different classes have different numbers of different cell types. And the more researchers investigate, they find specialized mechanisms that make different cell types different from each other. And I went into an argument that I don't have time to go into here, that would be beyond, that would require many different protein-protein interactions that would be beyond the edge of evolution. But also at that time, I said, well, there's pretty good evidence that you can, break a gene or you change a gene a little bit and come up with a new species. So somewhere then between classes and species was the edge of evolution. Somewhere, design is needed at least down to birds and fish and so on, and not so up to, brown mice versus black mice versus other things, different species in the same genus. But that was where the actual edge occurred could not be decided at that time. But science marches on. And we have a lot more data now than we had a dozen years ago when I was writing The Edge of Evolution. And we have enough to narrow the edge down even further, I think. And one of the very prominent developments is the development of new techniques for sequencing DNA and determining exactly what the change in DNA is that gives the change in the property of an organism that allows it to survive. What causes, say, a finch beak to be bigger or smaller? Nobody knew, say, a dozen years ago. All they could say is that, well, these finches with bigger beaks, they survive better under these circumstances. But you say, why? What caused that to change? I don't know. But now we can tell, it turns out. Now, I can't tell, but other people can tell. And one of the surprising things that has become more and more evident in the last dozen years is that much Darwinian evolution proceeds by breaking old genes, by breaking or degrading things that were already there in the organism, not by constructing new systems, but by breaking old ones. And it's interesting to think of a few implications here. We can understand why breaking would be frequently seen, because disabling a gene is likely to occur hundreds of times faster than making a specific change in a gene. If you want to break your car, you could just take a sledgehammer and smack it anywhere. Pretty much a lot of hammer blows would do a lot of damage to your car. But if you wanted to make it go, instead of 100 miles per hour max, 200 miles per hour max, you'd have to make very careful and specific changes. Well, it turns out it's faster. And so if a mutation, even if it breaks a gene, if it has some helpful effect, then it will be selected by natural selection. Natural selection doesn't care whether it's building something or not. It just cares who's surviving and who doesn't. I will pass over these guys in the interest of time. Well, how can that be that by breaking a gene, it's helpful? Well, here's an illustration. This is a picture I got off the internet. And here are some soldiers here, and back there are some vans. Turns out this was a South American country somewhere, and turns out the Army had gotten a tip that a drug-growing and running operation was occurring on a farm up the road, and they went to break it up, but somebody got tipped off in the drug ring, and they blew up the bridge. Now, from the point of view of the drug gang, this is a beneficial change, isn't it? You blow up the bridge and you live another day. But this does not tell you how bridges can be built. Bridges can't be built by any process that looks like this. Nonetheless, this is a change that helps. And it turns out that there are things in biology that are almost exactly like that. Malaria, again, it turns out the malarial parasite has to bind to specific proteins on the surface of the red blood cell before it can go inside. One population in Western Africa has a mutation which breaks its ability to express a protein on the red blood cell that malaria binds to. It blew up the bridge. It threw it away. Now it can, presumably that protein was doing something helpful, but apparently it wasn't as helpful as, as avoiding being eaten by a malarial cell. So this is a wonderful illustration of Darwinian evolution. And here I've got a table of the handful of mutations that are known to be helpful in malaria. And here's sickle cell, which I won't go into now, but all of the other ones involve breaking or degrading genes. Malaria, no, that's a parasite. But it's not just malaria and parasites. Lots of things can be broken to be helpful. A story in the New York Times from a couple years ago reported on a study that examined people who drank a lot of milkshakes, but their cholesterol levels never went up, and they never had heart attacks. The family history, they never had heart attacks. It turns out there was a specific gene that was mutated. What did the mutation do? It destroyed the gene. It destroyed it. It was reported a few years back that some horses, I didn't know this, I'm not a horse guy, some horses can trot more smoothly than others, and they're preferred by riders, and so we humans have bred these horses far and wide. It turns out the mutation that allows for a smoother ride is a nonsense mutation. What that means is it breaks the gene. Polar bear evolution. Polar bears are supposed to have descended from grizzly bears or brown bears. Even the polar bear genome has been sequenced. It was sequenced a few years ago, and the authors say, we predict that a large proportion of the mutations would be functionally damaging. The most highly selected mutation was one that broke a gene involved in fat metabolism. Fat metabolism is very important to polar bears because they live in this cold environment and have to insulate themselves. The long and the short of it is that almost every example of evolution you have seen is a result of a broken gene. All the varieties of dogs, a bunch of different breeds of dogs have had their entire sequence, entire DNA sequenced, and the important mutations are loss of function ones. Here's a man named Richard Lenski who does wonderful work at the Michigan State. He has been growing a culture of bacteria for 20 years, and each day he takes a flask of bacteria, he takes an aliquot from it and puts it in a new flask, new broth and stuff, and lets it grow there. And it grows about 7 generations per day because bacteria reproduce quickly. And that was 20 years ago. Turns out they've gone through about 60,000 generations so far. And it turns out he published just a year ago a list of the most highly selected genes from his experiment. They were all loss or degradation of function. And I wrote a review of some of this stuff a few years back, and I made up something. Because if you make up a really cool slogan, people remember it, and they have to deal with it. And I came up with what I called the first rule of adaptive evolution. And it's this: break or blunt any functional coded element, like a gene or a control region, whose loss would yield a net fitness gain. If you think about it, hey, if some gene it would be better to lose a particular gene. That's easy. Darwinian processes can do that in a snap. There's lots and lots of damaging mutations that could occur. And here's a little example. So mutations at many different points might break a gene, but if you're trying to build something, mutations at a couple of different places would take a very long time, and there aren't very many of them. All right, I'm going to skip this. Okay. It turns out that I think Darwinian evolution is self-limiting. That is, 3 factors restrict Darwinian evolution at higher levels of biological classification by actually promoting it at lower levels of biology. And the gist is this, that organisms adapt most quickly by degrading genes that are already there. But over time, that limits the repertoire of organisms, and they get less flexible, and they get stuck in their current environment. And the three factors are random mutation, natural selection, essentially the pieces of the Darwinian mechanism itself, and irreducible complexity. I don't have time to go over exactly what's going on, but the family line is a cute little slogan I met to emphasize that many species that are pointed to as wonderful examples of evolution that you'll find in biology textbooks, like the Darwin's finches in the Galapagos Islands. Everybody's heard of those. But if you think about it, if you classify the Galapagos finches, they have the same 1, 2, 3, 4, 5, 6 higher biological classifications, and they differ from each other only in the two lowest classifications, species and genus, from each other. And yet these things are supposed to have been undergoing intense selection for 2 million years since they arrived at the Galapagos, whereas that's the time estimated. Now, what's this family? It turns out there are, 1, 2, 3, 4, 5, 6, 7, 8 levels of biological classification. But as you go from level to level, the differences decrease a bit. Different domains, different kingdoms, plants versus animals, different classes, birds versus fish. Well, when you get down to species, you have one kind of finch versus another kind of finch. So an analogy I made up was, well, that's the family line right there, is that you can think of biological classification, those 8 different levels, as the place values, an 8-digit number with 8 different place values. So you have the cents, the dimes, the dollars, and so on, up to hundreds of thousands of dollars. So with the Darwin's finches, they have changed only in the first 2 columns of this 8-digit number that I put in terms of money just to make it feel more realistic or have a bigger impact. And over 2 million years, they've turned to this 8-digit number. So the changes they've undergone are biologically trivial. Oh, there's the family line again. I'm going to skip this. Another great example of evolution that's been pointed to repeatedly are things called cichlid fishes in the Great Lakes of Africa. And it turns out that some fish in Lake Victoria have only evolved in the last 15,000 years. People know that because they say 15,000 years ago there's evidence, geological evidence, that the lake dried up. It's fairly shallow. And yet it contains now species that are found nowhere else in the world. So the conclusion is those species must have evolved right there. And this is trumpeted far and wide, put in biology texts and so on. But if you compare the species across different lakes, they are pretty similar. They settle on very similar shapes and so on. So there seems to be a very limited diversity that can be achieved through this. What's more, I'm going to skip this. If you look at cichlids, cichlid is the family, and only new genuses and species have turned up in all of that time. Lake Victoria is about 15,000 years old. The other African lakes to which their fish are similar are millions of years old. But just like Darwin's finches, the change is confined to the sense level of that figure, that $100,000 figure. And a friend pointed out to me that he says when you file your taxes, the IRS tells everybody to round off to the nearest dollar their figures. And if that were followed in biology, all of this vaunted difference would be disregarded. I got lots more of examples of them too. It's not just fish and birds, it's reptiles and flies and beetles and plants and everything that's been looked at so far. So I have revised the edge of evolution down from classes to about the level of families. However, I raised it and say genera seem to be able to be produced by random processes. Well, what's the difference in families? Families are the level of dogs versus cats versus bears and so on. At that level, my argument is these required separate design. How that design was affected is a different question, but they required separate intelligent input to get these differences between them. Okay, I'll end it now because I want to have time for some questions. Let me just reiterate that surprisingly not everybody agrees with me, and I have at these places put up some responses to criticisms that these ideas have received. So if you want to get into the weeds with it, you should go there and look them up. So thanks very much for your attention. If anybody has a question, we can go with it. So
we have time for questions.
Okay.
About 7 or so minutes. So please ask away.
Yes, ma'am.
How long ago do you think this event of design happened based on?
The short answer is I don't know. But a different answer is that I myself think that, say, the Earth is very old, billions of years, and I think common descent is probably true. I'm not wedded to any of those ideas, but as I said in the beginning of the talk, I think those points are comparatively trivial. It's not, how long ago life was made. It's whether life was made intentionally or not. And that's the most important philosophical, scientific question. So that's what I concentrate on. Oh, come on. All right. Yes, sir.
Your colleagues in the scientific community, have you converted any of them to what extent have you converted them to your point of view?
Well, it's kind of interesting that there have been some folks who were very sincere Darwinists and have changed their mind. But since they are still in academia, it's because they haven't told anybody. In the beginning, in 1996, when many of you weren't alive or didn't drive, then a lot of scientists and a lot of folks were kind of intrigued by this idea. And we're saying, hey, that's weird. Yeah, it's interesting. And what is this about? And they say, oh, I have to think about that. Nobody says that anymore because beginning about 10 years later, mid-2000s or so, design got much in the news, and some parents of students wanted these ideas to be presented in schools, and people freaked out, and the scientific community was whipped into action, and they put out all sorts of publications. They gave all sorts of talks to stop this menace. And so since then, most scientists will not express even intrigue with this idea. You cannot say casually, if you have a job in academia, that, hey, I think there might be something about this intelligent design. I mean, maybe not everything, but I think there's something about it. You be out on your ear, especially if you don't have tenure yet. So it's kind of actually decreased, which is a drag. But I don't care, because the science has gone exactly the opposite way. The more we know, the more blatantly it points toward design, and the more it shows that Darwinism can't do things that even I thought it might be able to do 20 years ago. But even I, now think that even comparatively trivial things are beyond its capacity. Yes? Oh, I'm sorry. Go.
With your colleagues not believing anything about what you're saying and are very negative against what you're saying, I'm curious of how you keep your job.
Well, it's called tenure. Hey. Yeah, it turns out that's one of the reasons tenure was invented. Yeah, it turns out I had tenure before I got involved in this controversy, and so they're kind of stuck with me. I imagine you could. You could, if you were willing to say, well, this guy clearly has lost his mind because he's talking about this stuff, so he can't teach, he can't teach impressionable undergraduates. But they haven't done that. Lehigh's been comparatively good. A number of colleagues I know that are interested in this have been jumped on much harder than I ever have. They've lost jobs, students have been kicked out of labs, people have been denied tenure. So there's a lot of emotion, around this topic. Yes, sir.
Are you allowed to talk about intelligent design in your class?
No, I'm not. The department has a rule saying that no faculty member, in case anybody else wanted to, no faculty member can talk about intelligent design. However, if a student asks a question, then I am allowed, but I cannot initiate discussion of that. But I don't care. I'm not trying to convince, the faculty at Lehigh, or I want to convince the broader public. Yes, sir.
So I believe you've probably heard these criticisms before, but some arguments I've read in certain evolution textbooks is that a lot of these systems that are claimed to be irreducibly complex are actually can be actually reduced. You can knock out certain proteins and they can still function. Like such as, I forgot what specific protein they knocked out, but I believe there was a protein they knocked out in the flagellum and it still remained functionality.
Well, yeah, I mean, a number of systems have their function and then kind of have decorations and stuff, things that are not really required. So you can say, well, how about the cheese on the mousetrap? Does it need that cheese? No, it still can work, but whether or not the cheese is dispensable, the other parts aren't.
So you're claiming that it's irreducibly complex, or you can reduce these systems to a certain point.
Some of them, yeah. At least that's what I think now, with the evidence we have available. Let me give you an example. Turns out that the flagellum, like I put up on the screen, there are variations on the flagellum. The one I showed you is from a bug called E. coli, which is a common bacteria. But there are other flagella which are essentially bigger. They put out more force. Sometimes there's multiple flagella which spin together and so on. And that has been used as an argument in intelligent design. You say, hey, you say evolution can't produce the flagella. Look, it produced dozens. And you say, what? Where did you show how it was produced? And they say, yeah, but no designer would make all these variations of a flagellum. And you say, okay, then show me how you convert one to another. Nobody's ever showed that even starting with a flagellum, you can get to a distinctly different one. There's a flagellum, I have a slide of it somewhere, discovered in some bug that has a dozen or so flagella packed together, and in the interstices of where they are packed together. If they rotated together, when they were packed together, they'd smack against each other and wouldn't be able to work. But there are proteins that act as interstitial gears that counter-rotate as the flagella go one way. So this is the kind of thing they're saying, this disproves irreducible complexity. This disproves intelligence. No, it makes it more difficult. Intriguingly, it says, did design extend much, much further than we give it credit? Or even these variations, in flagella and other stuff, are they beyond Darwinian processes? And right now, yeah, I think they are. So this has a lot of implications for how deeply into nature design extends. And one other point worth making is that the response to this, as I said, is that no designer worth his salt would, are you saying it puffed into existence all of these things? And so my critics retort with a theological argument. These are the guys that are championing science, and they essentially say, no, these weren't designed because we don't think God would have done it this way. It's silly to think he cares about this variation on this flagellum and this one on that one. But they haven't done any scientific work to show even, modest variations between different kinds of flagellum are explainable by Darwinian processes. Almost all of this is assumption. Yes, sir?
Well, hold on. We're going to have to end formally here so that the students can go because their time is up. So let's thank Dr. Behe one more time. And feel free to stay and ask more questions for a little bit longer, but formally we are ending.